want to start by appreciating the AfriScience Network for doing me the honor of inviting me to give this talk. Can you hear me, please?
Yes, we can. Please, just go ahead.
Okay. I bring you greetings from the Federal University of Ekiti, the university that is most sought after in the country today, ably led by Professor Abayomi Fashino, our indefatigable Vice Chancellor, who we call the Ekweme One of Fuoye. They all bring you greetings. I also bring greetings from the Biotechnology Society of Nigeria. I have the honor to head at this point in time. I'm here today to share with you what I have gathered over the years about biosafety evaluation or assessment of genetically modified foods and crops. I'm going to introduce the matter, talk about the safety assessment paradigm, containment in the laboratory, confined field trials, risk analysis, risk assessments for general release, and under that we will discuss food and feed safety and environmental safety. Then we'll talk about risk management, which is a part of risk analysis, and then risk communication, which is also part of risk analysis. And before I begin to do that, I want to reiterate that all scientific academies of the world agree that approved GMOs are safe. Let me tell us a little about the advantages of GMOs. They've been modified to resist biotic and abiotic stress, such as insect and drought resistance. They give high yields. They give increased flavor, nutritional value, such as we have in golden rice. The telomerase that's just been released is resistant to insects and resistant to drought. GMOs help us in soil conservation due to use of herbicides, and there is no till farming with herbicide-resistant crops. And GMO foods are easier to grow, they are safer to grow, they are safer for the farmer. It gives us healthier farmers, richer farmers, and food is cheaper using GMO technology. Despite all these apparent advantages of GMOs, some people have concerns about GMOs despite their great advantages. The main concerns are to do with toxicity, allergies, cancer, environmental issues, which may affect the consumer. And they claim that while current research suggests that there are few risks with this technology, they believe more long-term research is needed. Well, to talk about biosafety then, to address the concerns of these few people, we want to tell them that biosafety has been established a long time ago to regulate GM technology and make sure that it is safe. I'll tell you a little about the birth of GMO biosafety. It has to do with a conference that was held in Asiloma in the early 70s. You know, after the discovery of the structure of DNA in 1954 by Watson and Crick and Franklin, there was about a 20-year lull and nothing happened. Why? Because the scientists didn't know how to cut this featureless molecule that goes on and on and on, GGC, TTC, BGAT, on and on and on. They didn't know how to cut and join it. But in the early 70s, suddenly they discovered the enzymes to cut and join DNA. And the restriction enzymes were used to cut DNA and the ligases to join it. So a sudden burst of activity started, and the scientists were like children who just got a new toy, and they were eager to try it out. In 1974, Paul Berg and other scientists recognized the potential of recombinant DNA technology, and they now saw that it has potential for good and harm. What happened? Paul Berg was working in his laboratory using this new he caught up the virus, monkey virus, SV40 monkey virus, which causes tumors in rats. And then he also caught up the bacteriophage. That, hello? Can you hear me? Can you hear me? Okay. So he caught up the SV40 virus that causes tumors in rats, and also caught up bacteriophage that attacks bacteria, and joined a piece from the SV40 and a piece from the lambda phage together, and was about to put it into E. coli. Paul Berg, by the way, was the very first person that made a recombinant DNA molecule. So, as he was about to put it into the E. coli, his colleagues appealed to him not to do it, because if he puts it into E. coli, anybody in the lab that comes in contact by mistake with the E. coli will go ahead to develop tumors and come down with cancer and die. And it was at this point that they recognized that this technology had great potentials for good and also for harm. At that point, scientists all over the world agreed to halt all experiments and research using recombinant DNA molecules and the DNA technology. And they wrote to the Academy of Science, stressing the need to regulate this technology. So this led to the Ashiloma, famous 1975 Ashiloma conference, at which all these international scientists decided on the guidelines to regulate GMO technology. And when they had made up these guidelines, everybody was happy, and they now went and continued their recombinant DNA work. So a lot of people do not know that biosafety was actually established by the scientists who developed recombinant DNA technology. This is the man, Paul Berg, who started it all, the first to make a recombinant DNA molecule, and who drove the action for establishment of guidelines to regulate GMO technology. He also, of course, got a Nobel Prize for his work for chemistry. So all we're going to discuss today derive from the guidelines from the Ashiloma conference. So the biosafety of genetically modified organisms refers to policies and procedures adopted to ensure that GM organisms are safe for humans and for the environment. You know, the world population is going to increase by 70% by 2050. Where are we going to get food to feed all these people? Biotechnology is being utilized to produce genetically modified crops to help provide for the needed increase. And while doing this, the overriding consideration is safety. So, during the development of early GM crops, therefore, a multi-component safety assessment paradigm was developed by scientific and regulatory authorities such as OECD and WHO, FAO. It is called the Cartagena Protocol on Biosafety. It is applied to individual GM food crops to determine whether they are as safe as foods from non-GM crops. In all countries, there are signatories In all countries that are signatories to the Genetic Protocol on Biosafety, environmental risk assessment is required for the regulatory approval of GM organisms. Environmental risk assessment includes both food and environmental safety. So there is regulation over all stages of permitted use of GMOs, from the laboratory to the screenhouse to the greenhouse to confined field trials to general release or commercial release and post-release activities. Let's start at the laboratory stage. At the laboratory stage, it is here that the safety consideration starts. Safety is ensured at the laboratory level by containment, use of hoods, negative lab pressure, so that even the air from the lab is not allowed to come out and mix with the air outside. The first principle for dealing with potential risks in the guidelines from the Ashiloma conference was that containment should be made an essential consideration in experimental design. And the second principle was that the effectiveness of the containment should match the estimated risk. So if I'm not using, for instance, a microorganism that is harmful in any way, I don't need to contain the lab so much. It boils down to killing an ant with a big hammer. So that's one of the things they also decided there. So in each country, the competent authority enforces this international agreement. In our country, it is the National Biosafety Management Agency, NBMA, that is in charge of this. So if you are going to carry out any recombinant DNA experiment, say you want to make your own GMOs, you first have to make sure that your university has an institutional biosafety officer. And this is the first person you speak with. You discuss your intention, the work you want to do on paper. So he evaluates it on paper before it enters the laboratory at all. And thereafter, you invite the NBMA to come and certify your lab for recombinant DNA work before you now start. After you have done your work at the laboratory level, screenhouse level, greenhouse level, confined, you will now carry out confined field trials. That's containment again. The confined field trials, or CFTs, are small-scale field experiments to address biosafety requirements and evaluate the performance of specific traits in genetically engineered plants. They are conducted to collect biosafety data for presentation to regulatory authorities, in our case, the NBMA, to produce plant material for further studies on food safety. All the material you will use to evaluate food safety is produced during confined field trials. It is also conducted to evaluate gene expression in the field and test agronomic performance, select the preferred lines, and breed the new traits into farmer preferred lines. If you make a wonderful crop and you do not breed it into the lines that farmers prefer, you're on your own. The farmers will not plant it. Believe it or not, forget propaganda, the farmers have a mind of their own. If they don't like your product, they won't grow it. So safety at this level is also ensured by containment. After the confined field trials, the next level is now the level of general release. And by international agreement, countries usually have a legislative scheme regarding the use of new technologies and or their products. For biotechnology, the Cartagena Protocol on Biosafety states that before a genetically modified organism is released into the environment, a determination of the possible associated risks to the environment and to human health should be undertaken. Therefore, genetically modified organisms always undergo a very severe regulatory process. So risk analysis is a regulatory framework to assist the decision-making process for the possible permission to use a biotechnology product. This risk analysis involves three things: risk assessment, risk management, and risk communication. Risk assessment evaluates the risks of GMOs to human health and the environment. It is determined by evaluation of data contained in a dossier submitted by the developer to the regulatory or competent authority, in our case, the MBME. So the risks here may be direct or indirect, immediate or delayed, which decide the deliberate release of GMOs and what the risks they may pose. So, two things are involved in this risk assessment: food and feed safety and environmental safety. Food and feed safety is what we mean when we say effect on human health. So how do we assess food and feed safety? Safety assessment of GMO crops involved two things: the assessment of the pure protein you have put into the crop. That we regard as the intended effect, because that is what you set out to prove. The second is the assessment of the whole foods from the GM crops. It is also known as the test assessment of the unintended effects. Now, how do we assess for the expressed protein, that is the intended effect? We do so in two ways. We assess for allergenicity, we assess for toxicity. How do we assess for allergenicity? An allergen is the abnormal reaction of the body to a previously encountered allergen introduced either by inhalation, injection, skin contact, and the symptoms are itchy eyes, runny nose, wheezing, skin rash, or diarrhea. An allergy develops when the immune system becomes hypersensitive to a food. Majority of the food allergies are attributed to a relatively small amount of small number of foods, such as fish, soybean, peanuts, tree nuts, oysters, and so on. So the foods that contain allergenic proteins are known to science, making it a little bit easier for us. The potential for allergenicity is assessed for in the new proteins in GM crops to ensure that they are not similar enough to allergens to cross-react with antibodies that are present in the bodies of people who are who have food allergies. The key components for allergenicity assessment, number one, is the history of safe use. The source of the protein is an important consideration in safety assessments, which we call the history of safe use. That is the organism from which you borrowed the gene that you transferred to the GM crop. Is it a safe organism? The second is that the transgene must not originate from a source that is known to contain an allergen, a known allergen, e.g., groundnuts, peanuts. Peanuts are known to contain allergens. Therefore, if you are making any genetically modified organism food, you must not take genes from the groundnuts. So let us look at some of the things on the market. The BT crops contain the cry or crystalline proteins from Bacillus thuringiensis. Bacillus thuringiensis is a soil organism with which we have interacted with over the centuries. So the Bacillus thuringiensis, it has a history of safe use. For herbicide tolerance crops, the CP4 agrobacterium strain, from which we borrowed the gene to produce that produces EPS-PS proteins, they do not produce proteins that are known to be allergenic in humans. They are also organisms that we have mixed with and interacted with over the years. So that particular strain of Agrobacterium is the CP4 strain, also has a history of safe use. So the history of safe use is also necessary for any intermediary hosts you are going to use. For instance, in E. coli, that is always, E. coli is the workhorse of molecular biology, of genetic engineering. You use it because we know all about it. It is easy to grow, it is harmless, and so we use it to sometimes multiply genes, and you use it as hosts for a lot of things. So E. coli, for instance, it lives in our guts, in the gut of human beings. So we have associated with it for millennia. So it is an organism that also has a history of safe use. That's why we can use it. So the second thing we consider in analyzing for whether this crop has the danger of becoming an allergen or triggering off allergic reactions is the amino acid sequence of the new protein. You check the amino acid sequence of the new protein and compare it on the database that contains all known allergens in this world, and compare for similarity of the amino acid sequence of the new protein with the amino acid sequences of the allergens that are in that database. So you use bioinformatic tools and bioinformatic softwares to do this. And I've just the database where you find all allergens in the world is here. So when you do that, you do this alignment, any new protein that has an amino acid similarity of 34% or more over any 80 amino acid sequence, or has any 8 contiguous amino acid sequence in common with a known allergenic protein, is considered to present a risk of triggering an allergic reaction and must not be used to make a genetically modified organism. Okay. Another factor that we consider is the resistance to digestive enzymes. If your new protein is resistant to peptide, the digestive enzymes that our body uses, it's a red flag that protein, you know, is safe to eat. All these methods together have been very effective at identifying potentially allergenic proteins. Insecticidal and herbicidal tolerance proteins in current use are degraded rapidly by digestive enzymes. After more than 25 years of use, no approved GM food has been found to induce allergic reactions. Mark my word of the phrase approved. No approved GM food has been seen to pose this risk. The second item we said we test for in checking the safety of the expressed protein is toxicity. The tests for toxicity are similar to those for allergenicity. You check the effect of digestive enzymes on them. You check the bioinformatic similarity of the amino acid sequences. But in addition, for toxicity, you also carry out in vitro acute toxicology studies, to assess for toxicity potentials. The difference between the bioinformatic studies for toxicity and allergenicity is that while for allergenicity, we have a dedicated database for all allergens, to compare with. For toxicity, you just compare with the sequences of all proteins in the NCBI database, and to check if it matches with any of the proteins in the NCBI database, then you check the history of that match and know whether it has a history of toxicity or not. In many cases, active toxicology studies are carried out in lab mice using proteins isolated from heterologous expression systems such as E. coli following OECD guidelines. The thing is that if you want to get the express protein from the food, like from BT cowpea or from BT corn, you will really need a lot of corn or cowpea to get enough to run your toxicology studies. That's why it's often derived from expression systems such as E. coli. That is, you insert the gene into E. coli, and then E. coli makes the protein for you. That way you get enough protein for your rat studies, for your animal, for your mice studies. Scientists will then calculate dietary exposure to the protein, like how much can a normal average human being consume, in a day, 2 or so. And when they have calculated that, you multiply it by 1,000. That is what you use to feed the lab mice. Can you see the safety, the large safety margin, So, also, the European Food Safety Authority, a lot of people will tell you, oh, Europe has banned GMO, but they only need to go to the EFSA website and see the activities there and all the food that's been approved for use in the EU. The EFSA requires a 28-day rodent toxicology study with the pure protein. Having done all this, none of these studies have shown adverse effects for any protein, or GM cross for toxicity or for allergenicity. So we have just finished with the safety assessment of the expressed protein. That is the intended effect. The next one is safety assessment of the whole food. Safety assessment of the whole food is assessment of unintended effects. That is to say something that happened that we didn't plan for. You see, no food is absolutely safe, but the foods we eat, we say they have a history of safe consumption. So we compare the genetically modified foods with foods that we consider to have a history of safe consumption. So in GM food safety evaluation, the objective is to show that the GM food is substantially equivalent to foods from closely related non-modified crops that themselves have a history of safe consumption. How do we evaluate the safety of the whole food? For the safety of the whole food, we look at two things: molecular characterization and compositional makeup. For molecular characterization, this is checking the integrity of the inserted DNA, the copy number, to confirm that the insert did not disrupt any genes or create any new open reading frames or insert any DNA backbone. You see, the thing is that during the transformation to make these GMOs, the insertion is random. So the new gene can insert anywhere. And sometimes when such an insertion takes place next to another gene, that insertion, that activity silences that other gene. So that's why we're interested in that. So these elements are associated with the safety of the products because unintended effects can occur from transformation. The thing is that during confined field trial, a lot of the events, what we call events, is a plant that grew from insertion at a particular nucleotide. That is, like you say, between nucleotide 104 and nucleotide 105. That is an event. So all the events that inserted in difficult places that will affect the plant have been noted and such plants removed during confined field trial. So that's why, I will tell you, oh, they have not found any adverse effects So you can believe it, because that is because they have already removed a lot of such plants during confined field trial. The second thing that we look at in analyzing the whole food is the compositional analysis. There are two ways of doing the compositional analysis. You can have a targeted approach or a profiled approach. The profiled approach means you analyze everything by all the components, they all metabolized by metabolomics, proteomics, transcriptomics, genomics, everything. You analyze everything and you compare. That's a lot of data to generate. And good science does not believe in generating data for the sake of generating data. You generate data that is useful. So the targeted approach, therefore, is the favored approach nowadays. Why is it called targeted? It means that you analyze the relevant metabolites, the ones that are of importance in that particular crop. So the first thing you do is a general analysis, proximate analysis. After the proximate analysis, by the way, analyzes the protein, carbohydrates, fat, ash, moisture, such big nutrients. And then after that, you now go to a little specialization. For every food, the OECD has a list of the components you must analyze for that food if you are doing anything with it and you want to find out if there is an effect. Take, for instance, legumes. Legumes have oligosaccharides that give you rumbling tummy when you eat them. They have anti-nutrients, lectins, all that. So if you are genetically modifying a legume, when you are carrying out compositional analysis, you must analyze for those components to know, has it increased, has it decreased? That is what is done. If you are modifying something like cassava, you can imagine that the cyanogenic glucosides of cassava will be your target after the first set of analysis. You will go and check. Did it increase? Did it decrease? Sometimes you find out actually that something good has happened, that a bad component has been reduced or have been removed. So that is how you do it, and then you compare what you get with the levels in an isogenic line. That is a non-modified, really close relative of the GM food. So whole foods from controlled field trials in which the GM crop and is closely related non-GM control, that is the near isogenic line, as well as several unrelated commercially available non-GM varieties, they are grown together and then they are the ones analyzed in this compositional analysis. Also, apart from comparing the GM food with the non-modified near isogenic line, you will also use published chemical compositional data to compare. And there are various websites on the internet, like the one run by ELC, where you have published data for food. You also use that to compare, because you could compare a GM and the non-GM counterpart, and you find that, okay, maybe an undesirable characteristic is high in the GM, or a desirable characteristic is low in the GM, and you want to disapprove it. Before you do that, you must go to published literature and make sure that that value you got, that it is outside the general range obtainable in literature. Once it is obtainable in literature, then that product is good to go. So from all the compositional studies so far, over to 25 years of consumption of genetically modified foods, there has been no significant adverse unintentional compositional changes observed in approved products. This shows that in approved products, the process of transformation and expression of proteins in GM crops has not led to adverse unintentional effects in the products chosen for commercial release, and that therefore these products are substantially equivalent to their non-GM comparators with a history of safe use. Now, when compositional analysis has shown equivalence and there is no scientific, then there is no scientific reason for carrying out animal studies. However, if assessments based on molecular, biological, chemical data does not allow an informed judgment on safety, a conclusive judgment to be made, then animal feeding studies are indicated. This means that toxicological studies on whole foods are only recommended on a case-by-case basis, where the compositional studies leave cause for concern. The studies are comparative in nature, and as they are not pure chemicals, but mixtures of components already found to have equivalence by composition. And the pure transgenic protein is not toxic or allergenic. One of the feeding studies often conducted with whole foods from GM crops is the 90-day feeding study in laboratory rats. It often includes multiple reference groups of rats. Why the multiple reference groups is that when you have compared the GM food with the non-GM food, you will also compare the results with these reference groups of rats that you have fed completely unrelated feed fractions, different from the near isogenic lines, so that you make sure that you don't reject a food because it had values that already occur in commercially available non-GM plants. The reference groups are very useful, therefore, for resolving such statistically significant differences between GM crops and non-GM crops. You might have such differences and they will not have biological relevance. So the conclusion from feeding studies with whole foods is that there is no evidence of adverse effect, and exposure to long-term feeding diets have found no adverse effects. In addition, many people carry out livestock studies with poultry and broiler, but these are really not for safety, but just to check nutritional performance in such livestock. One more thing that is of concern about the food safety is horizontal gene transfer. The horizontal gene transfer is stable transfer of genes from one organism to another. That is, people are afraid that with the transgene enter my body if I eat it. And there is concern also that, oh, could the recombinant DNA not be transferred to cells of consuming animals and humans, especially in the gut? But studies have shown that studies have shown that this does not happen, that the body just metabolizes the DNA as if it is coming from the other foods you ate that are not GM, because all food contain DNA, and the body does not segregate the DNA and take the one that is GM and use it for something else. It degrades all of them during digestion. So studies have shown that this horizontal gene transfer is very, very low. They don't see it in blood samples, they don't see it in tissue samples, and that's why we feel we are good to go. Another horizontal gene transfer concern has to do with antibiotic resistance, because during the experimentation to make a genetically modified GMOs, that we use antibiotic resistant genes as markers. A marker helps to tell you to tell you what you are doing, to show you where your gene is. So the people are saying that these antibiotic resistant genes could enter the bacteria in our gut and make them antibiotic resistant, and therefore they become super bugs, you know. And it has been shown that this transgene does not survive digestion. It is digested just like all other DNA that is in the other food that we eat. And the antibiotic resistant genes used in genetic engineering are found naturally in many bacteria. So it's nothing new. You know, even some bacteria in our guts have them. The bacteria in the soil have them. The bacteria everywhere have them, because the bacteria use it to outcompete their fellow bacteria in the environment for nutrients and this stuff. And thirdly, antibiotics whose resistant genes are used for genetic engineering are usually those that are not often prescribed. I don't know how many people here have ever been prescribed kanamycin for i ailment. It is a seldom used antibiotic. And also, antibiotics that have many alternatives are also used, like ampicillin. Ampicillin is an antibiotic that has many alternatives. So that concludes, therefore, examination of food safety. And it has been found, all these things found, that food safety is ensured, and all genetically modified foods that have been approved for human use are safe. They are not toxic, they are not allergenic, they don't transfer antibiotic resistance to you, they don't enter your blood and your tissue, and so on. Then the second aspect, you see, remember we said we are analyzing the GMO for safety to human health, that is food safety, and safety to the environment. Now we go to the safety to the environment. When we are assessing a potential or safety, potential or direct effects to the environment, it can be direct, it can be indirect. The direct effects, potential effects to the environment, have to do with non-target organisms. And the second has to do with gene transfer or gene flow. When we assess potential or indirect effects, we are looking at weediness, invasiveness, biosafety, biodiversity, and agricultural practices. What is effect on non-target organisms? That is the direct effect. Some people argue that if this insecticidal protein in BT crops, for instance, if it can kill the organisms that attack it, such as fall armyworm and so on, then it can kill butterflies and bees and all that are beneficial insects to us. But the truth is that the non-target organisms, because they don't attack the crop, they don't get to ingest the insecticidal protein. And the studies have not been able to find any adverse environmental impact of GM crops on the abundance of non-target organisms, especially when you compare it to the devastation on non-target organisms done by insecticidal sprays, as used to be before the advent of genetically modified insect-resistant crops. So, rather than have adverse effects on non-target organisms, it actually has beneficial effects on non-target organisms. If you come to a field and spray, all the bees and butterflies and everything will die. But with GM BT crops, no non-target organisms are not affected at all. Only the ones that attack the crop are affected. I know some people might also say that man is also a non-target organism. Yes, it doesn't affect us. Why doesn't it affect us? For two things. One, the insecticidal protein that protects BT crops, it requires an alkaline environment to work, to exert its biological activity. And the insect has that alkaline gut. We don't have. You all know that hydrochloric acid is the thing that is poured into our stomach when we eat, that our stomach is acidic. So it cannot exert biological activity in our gut. Secondly, the insect has the appropriate receptor for that protein for it to be processed. The human gut doesn't have it. So when that insecticidal protein gets into our digestive system, it is processed and digested like any other protein, because it cannot exert biological activity in the human gut. Now, the second consideration for direct effect in the environment is gene transfer or gene flow. It is argued that transgenes from GM crops can be transferred to other plants through polling by outcrossing, either to related crops, to weeds, or to wild relatives. There is evidence, true, that this can happen, but there are ways of managing it. For closely related crops, all you need to do is to distance the genetically modified crop from the non-genetically modified crop, either in distance or in space or in time. So if you don't have enough land to distance your crop from the man who is growing non-genetically modified crop, no problem. Just wait for the man to plant. 2 weeks after, 1 month after, plant your own. His own would have been setting seed by the time your own flowers. So you have separated, you distance yourself from him in time. So there is no crossing, whether it's going to cross or not. For weeds, you also you separate the approved GMO from the weed, or you use a different herbicide from the herbicide that is protecting that herbicide tolerant crop to destroy the weed. If the weed has accepted or taken the herbicide resistant gene and has become a super weed in the farm, what you do is that you just use one of the other herbicides and kill the weed. In the case of wild relatives, that one is done by the approving agency, because one of the questions you answer in the dossier that you fill to submit to the regulating authority is there any wild relative of this crop in the area that you are going to release it? If the answer is yes, they will not approve it for general release. It happened somewhere in Canada for brassica or so, and it was when the approval was given, and they said you must not grow it in Quebec or so, something like that. And in the end, they really had to withdraw that product. So that is then how the matter of gene transfer is taken care of. As for when we are talking now of indirect effect, has to do with weediness, invasiveness, and then effect on diversity and agricultural practices. For weediness, the major concern is with the potential of the crops of becoming weed or to become invasive. The thing is that non-certified plants can also become like that. So the transfer of herbicide-resistant genes to wild or weedy relatives can give rise to super weeds. Quite all right, but I've already told you how you handle that. So the assessment, the regulator will consider if such wild or weedy relatives are in the environment before approving the plants. So GM plants that show increased weediness or invasiveness are not approved by the regulatory authority because they will be a threat in the environment. You can see an invasive plant. You parked your car and you went on holiday and you came back and this is what you see. This is an invasive plant. A crop that is related to this or like this will not be approved for genetic modification. This is also an invasive tree or crop. It takes all the water in the environment, and if you are going to use it for GM modification, they are not going to approve it. We have a similar crop in our environment that looks like sunflower that must have come from the ports. You will find it on all the highways in this country. All the highways I know in this country, I find them. And once they grow on the roadsides, they enter the hinterlands and take over the whole place. Such an invasive plant will not be approved for genetic modification. Another indirect effect is the effect on biodiversity, that they might have indirect environmental effects as a result of changing agricultural practices that are required for that particular plant. These indirect effects may be beneficial or harmful. So for these agricultural practices and biodiversity, how do we address it for insect-resistant crops and for herbicide-resistant crops? For insect-resistant crops, the insect-resistant crops, they reduce the volume and frequency of insecticidal use. They cause less contamination of our water supplies and less damage to non-target insects. They cause a reduced pesticides use, which is generally beneficial to biodiversity, and less chemical and pesticide spraying means that we have a healthier farmer, a healthier environment, and healthier farm workers. For herbicide-tolerant crops, herbicide-tolerant crops lead to a shift away from toxic herbicides to safer ones. They encourage low-till agriculture, resulting in soil conservation. There is concern, however, that greater use of herbicides, even less toxic herbicides, will further erode habitats for farmlands, birds, and other species. Compared with conventional cropping practices, genetically modified herbicide-tolerant crops have effects on weed vegetation, with consequent effects on herbivores and pollinators that feed on it. What they are saying in this one is that a farm like this that looks so clean, because it is herbicide tolerant, so they have used herbicide to make it really clean. So it does not have those small weeds that you have when you weed manually. After a week or two, you now have a carpet of small weeds on the farm. They are saying that that carpet of small weeds is an ecosystem of itself, and that there are some ants and little bears and stuff that live there, and that herbicide tolerant corns have taken their environment away from them. Well, you decide whether you want the corn or you want to keep the small ecosystem of the ants for them. So after we have now finished with risk assessment, the next thing is risk management, to focus on did you find any risk? If you found any risk during all these assessments, what are you going to do about it? See, when we look at risk is equal to hazard times exposure. During the contained lab and confined field trials and all those, we were concentrating on containment. Therefore, exposure was 0 or near to 0. So we have reduced the risk by reducing exposure. But in the case of general release, exposure is maximum. It is 100%. That is why during general release, emphasis is on hazard, you must reduce the hazard to near to zero in order to have a low risk. So hazard identification and characterization have to be more complete during risk assessment because exposure cannot be controlled. During for insect-resistant crops, risk management is in form of gene stacking, building refuges, and good farm management. Gene stacking means that you have more than one gene, that is giving the insect resistance. In the teller maize that had just been released, if you look at the partner that is insect resistant, you will see that it has two cry genes, two crystalline gene from Bacillus thuringiensis. And so that if the insect is resistant to one gene, it will not be resistant to the other. The second method is to build a refuge around the GM farm that has non-genetically modified corn, so that insects that acquire resistance, when they go to that ring, they mate with insects that don't have resistance, and they lose their resistance. You also have good farm management to do this. In herbicide-resistant products, as I told you, you use the restriction of a growing area by approval and say, no, you are not approved to grow it in so area. And of course, the occurrence of super weeds is low. And thirdly, the development of germplasm collections, seed banks, tree nurseries, gene banks, by people like IITA, NAPGRAB, they conserve biodiversity so that the loss of biodiversity here is not there. Now, when you have finished all this, one of the things that the Ashidoma conference has put in place is that you must communicate with the stakeholders. You must communicate with the public. So the next thing is risk communication. All that you have been doing, have you communicated to the people? This is what we have done. This is how we have done it. This is how we normally do it. What I am doing now is what I am explaining to you, the stakeholders, how the regulatory system works. It is part of risk communication, so that we are, to a very large extent, all on one page about what is happening. And that is technology, and that is why intellectual property rights is very high in biotechnology, because there is complete disclosure. In conclusion, the new gene technology, has to be inspected, for its promise, from its benefits to human health and the environment. There is no evidence to prove that approved GM foods are unsafe. Instead, there is a lot of evidence to prove that it is very safe. Further research and observation should continuously be carried out as post-approval follow-up by both regulators and developers, independent scientists, to provide practical evidence of safety and benefit of GM technology. I really recommend that the research, in giving my recommendations, I would like to remind us that the research for all the GM products approved in Nigeria, the meetings for approval, the post-approval follow-up activities by the regulatory and promoting agencies are funded by international agencies. I think it's a shame for our country. Most ministries and agencies involved in science and technology, universities and research institutes, have no real budgets for research. It has been proven that any country which does not sincerely, massively fund scientific research cannot have development or innovation. So Nigeria, where are we? I have put some resource stuff for people who want to read further. As I say, thank you very much for listening.
Thank you.
Well, I'm sure that you will agree with me that most exhaustive in presentation, Professor Uzochukwu, who has tried to do justice to every aspect that one can see from the issue of the ecology, those that are involved in it, and those that need for actually push on regulation and various steps that are taken to ensure that for GM foods get to the market, they go through with the science. She has dealt with but about food, about the environment. She has dealt with regulatory issues, and she has reached some very fundamental conclusions that really engage the attention, so that when people in authority talk, they are talking from a position of knowledge, from a science-based position. So clearly there will be a vote of thanks at the end. At this point, that we are very grateful time to put this presentation to address the issues of concern, things that we have heard on television, on the radio. So we really thank you very much. Some questions and comments. It's 6:25. So I'm not exactly sure the best way to handle this, but I can see that a few hands had been raised earlier on. So may I request Adeshola Ajayi, if she's with us, to pose a question or comment. I've also seen Olugbenga Onoja. I'm seeing Prince Odo, and 7 more people who have raised their hands. We may not be able to take everybody. I'm sure the professor, we have her email. We may continue, and this is a very, very active platform on WhatsApp. I'm sure exchanges will continue. So for now, can we call on Adeshola Ajayi to ask the question or make the comments?
Good evening, sir. Thank you very much. I do not have a question. I mistakenly raised my hand while I was trying to type a comment. But well done, Prof. Thanks for the well-delivered lecture. Thank you, sir.
Thank you. Olugbenga Stephen, I know.
All right. Thank you, sir. Many thanks indeed, Professor Uzochukwu, for this presentation. I must confess that this is the first time I will listen to somebody who is quite proficient, knowledgeable, and intelligible in this particular field. All I've heard before are people speaking from the scratches. But you have really given us an in-depth, very deep angle to GMO foods tonight. Thank you very much. I am living here with so much knowledge about it. The truth is we cannot do anything against technology, but to move with technology. If you don't move with technology, definitely you'll be left behind. So I just have 2 questions very quickly. I wanted to ask, for a new technology like this, what is the post-approval monitoring systems that we are putting in place For example, I was part of the people that approved the vaccines during the COVID-19. All the vaccines, I was part of the approval. But one thing we did was to put in place a very strong pharmacovigilance to ensure that we monitor any adverse effect outside. So I don't know if there's a serious framework to contain it once, especially like the telomeres, once it gets into the system. Are we putting in place to ensure that we monitor any adverse effect? Then secondly, my second question is, we spoke about field studies. I wanted to ask, the field studies, were they done? Do we have any field studies done in Nigeria apart from the foreign ones? Because most of the times, we are in different climatic zone. For example, Nigeria is in climatic zone 4B. So the field studies done abroad you may need to domesticate some field studies to our climatic and environmental conditions here. So those are my two quick questions. But well done, madam. You have really made my day with this presentation. Big ups to you, man. Thank you.
Can I answer? Okay. I know there is a monitoring system in place. Anytime there is an approval, it comes with conditions for post-approval monitoring. And it is also there as part of the law, just as you talked about for the COVID. This is also part of the law. What the developer must report to the regulator, how he must report. In fact, there are forms he must fill for all those things, and even support that he must give to the farmer to make sure that the farmer grows. All this is part of the approval.
Excellent.
There's a lot of conditions for post-approval monitoring that is part of the law that NDMA implements. For the field studies, if you've been following the development of all these, you will see that the field studies for cowpea, BT cowpea, was done at IAR Zaria. The field studies for telomeres, the same thing at IAR Zaria. And the products that are in the pipeline, such as the rice that is in the pipeline, the newest rice, is being done by the Rice Research Institute, Badegi, I think. So it is completely homegrown. And that's what a lot of people sometimes don't understand. It's complete. They will tell you, oh, this big corporation, but it's coming from Zaria.
Thank you, madam. Thank you. Very well said. Thank you.
Thank you very much. So can we now move on to Uzochukwu Sunday Ayonda?
Thank you very much, Prof.
Yes.
Yes, I am here, sir. Yes, sir.
Please go ahead. Yeah.
Yes, I am Dr. Uzochukwu Sunday Ayonda from the Federal University of Ekiti. I must thank Professor Sylvia Uzochukwu and the organizer of this wonderful event. I must say that Professor Uzochukwu is an erudite scholar, and in our university we often call her Mama Science. Sometimes they call her Mama Scopus, because he has been mentoring many of us. I have two questions here. Why Prof was speaking the other time, she made mention that all the research that have been considered GMO to be safe. I want to ask if in all these researches, if accumulation over time were considered. For example, when we were young, we used to take, a lot of us, we take sugar, and then by the time we get to old, okay, don't take sugar again because it accumulates and cause diabetes. Is it possible for these GMO foods to later cause problem in future? So that's why I asked whether accumulation over time has been considered throughout all some of these studies. Another question I have here is that, is there any agency in Nigeria that certified GMO foods? And then, if there are the populace, are they actually aware of the non-approved GMO? Because if they are not aware, everyone will say, okay, GMO is this, is that, and is this. So I think there is a need for us to also know the non-approved ones. And then we also need an agency that will certify all these foods before consumption in Nigeria. Thank you very much, sir.
Thank you. Prof, please, can you address those questions?
Okay, Dr. Ayonda, thank you. You talked about effect over time, if it is considered. Okay. Effect over time, as I will say that genetically modified foods have come into their own because it's been consumed for more than 25 years now. So that, in fact, in their own rights, they should begin to be generally accepted as safe. So the effect over time, I think we've had time to see that, and we have not seen any of those people grow horns or anything like that. I think it has stood the test of time. And you said if there is an agency that certifies GM food, yes, I said it many times in my presentation that the MBMA is the one in charge of regulating and certifying anything to do with GMO in the country. That is the National Biosafety Management Agency, MBMA. It's in Abuja. It is this agency that everybody applies to, both for laboratory work, for the confined field trial, or general release, anything to do with GMO, you go to the MBMA. They are the agency in charge. And you said what of non-approved? Because I kept hammering approved. The thing is that it is against the law to bring to public use, commercial use, general use, any GMO that is not approved by the regulatory authority of that country. You cannot also import GMO that has been approved in another country to another country without the approval of the competent authority of that country. This is by international treaty. There was, before we got our biosafety law in place, before it was signed, it took a long time. People were importing GM corn into this country, but the agency in charge could not do anything because the biosafety law had not been passed. So it was not only until the biosafety law was passed, as soon as the biosafety law was passed, the ship that brought that corn, the one on ground that time, it was turned back to South America where it came from. And there was big lobbying everywhere, you know, because of it. And the regulator stood his ground and said, no, there is no approval for this product in this country. It may have been approved in your country, but it is not approved in this country. You cannot bring it in. So those importers started this big lobby to get that particular one approved. And it was done. It was finally done. It was approved. That was the first approval of genetically modified corn in this country. It was approved for import. So there is nothing like unapproved product. It must be approved before it will see the light of day.
Well, thank you very much, Prof. I think.
Thank you, Prof.
Yes, it's 6:37 p.m. We will take one more question. I know there are several. Can we share that with our presenter and amongst us later? For now, we will ask Prince Odo if he or she is still with us, or Prince, he is still with us, and
yes.
we'll begin to close. Prince Odo, are you with us?
Yes, I am still available, and I am so very grateful that I have opportunity to speak. I was so very sad if we have opportunity to speak. You began with a categorical statement, and towards the end, you also gave it. Your first statement was all scientific academies of the world agree that approved GM foods are safe. Inside your presentation, you said, mark my word, approved. And just about the end, you said, no evidence that that any approved GM food is unsafe. These are very categorical statements, and questions are so much that should be asked on that. Somewhere along your presentation, you talk about substantial equivalence. Now, before I ask my questions, which I want to dwell on this, there's a fundamental question I want to ask to precede these questions. And that goes to the title of the paper, assessment. Now, when one does assessment or regulation, there must be a standard experiment, a model, which will guide the assessment or the regulation. Let me go to the UK. When GMO was to get to the UK, the government said, look, we've never had food produced by this means before. This is novel. Therefore, we must undertake our independent research and assessment before we can accept it. And they put out not less than, they put out application for experts, and less than 30 applied, and Dr. Apat Pustai, I'm sure this name is familiar to many people, or some people. Dr. Apat Pustai came first, and he teamed with others in Rowett Research Institute to look at the safety of GM potato, which was prepared and given to him by the Scottish Agricultural Institute. And Apat Pustai expected it to be safe. His communication with me personally, I had direct communication with him, but he brought me to this research. He expected it to be safe. He said it was expected that he would get the Nobel Prize, and the Rowett Research Institute would be the center where all foods globally would be approved. But to his surprise, by 10 days of his research, the rats were dying when he did the studies. I won't go too deep into that. What is important there is this. In your first assessment, talk about proteins. Assessing proteins is not a sufficient means, because Apat Pustai is from Snowdrift, and for him, he had used it for many years. There was no problem. That was part of why he was shocked that at this time, using the same lesson, there was a problem. So why did it happen? He worked on it. He said he needed more funds, but right now all he could think about was that the method of application, genetic engineering, I call it genetic re-engineering, not genetic engineering, because the engineering is not from nothing. It was based on something that existed. So if something existed, you want to engineer that thing, it is re-engineering. It's not engineering. So my language is genetic re-engineering. So Apat Pustai concluded, look, it must be either the method or the tool or how it interacted, the interaction in the genome system. Let me cut it there. That now it is on the basis of Apat Pustai work that most EU governments have rejected them food. I said I had personal interaction with him on phone and GSM until he died, I think 2 years or 3 years ago, about, So now come back to Nigeria. We did not do that. We have not independently worked any GMO to independently assess safety or not. I don't know any. In my research work, I've done this since 1998. I've been talking to all the people, Professor Simon, when he was NABDA,
Please. The moderator, you have to mute this person. Mute this person, moderator.
Okay.
No, please. I'm the chairman of this session. You must allow Prince Odo to finish his questions and his comments. But Prince, can you round up so that we will ask Professor Uzochukwu to respond?
Oh, thank you so very much, Prof. You see, you're wonderful. You are wonderful. I don't mean to flatter you. Anyone who knows me for what I do, what I say. I'm saying that the issue is not a trivial issue because we're talking about human life. Animal life depends on good health. Good health depends on food safety, on safe foods. These three go together. Without safe food, you can't have good health. Without good health, your life goes to zero. That is death. So, we can't trivialize this thing. Thank you very much, Prof, for having time. So, let me go back. If you wanted to say, I also make a categorical statement. Prof, your first statement is categorically wrong. That is that whatever academy has said it is not safe. Your close to the end is also categorically wrong. Why? Because it has been shown that GM foods not safe. Even the first GM food, tomato, which was first studied by US FDA, they showed it was toxic. US FDA hid the results, and Alliance for Biointegrity used FOI to force it to bring it out. When it was brought out and it was tested, they found out it was toxic. And that was why FLU now banned it. That was between 1994 and 1998. It now banned it because of that. It was based on that grass, generally regarded as safe, came in to be used that the food will be tested again. After grass was rejected, substantial equivalent came in, which you quoted. Substantial equivalent is not scientific. It does not say that the food is safe. If there's substantial equivalent.
Prince,
yes?
Prince, because of our time, you've made your point very, very eloquently. So can we just ask Professor Uzochukwu to just briefly respond to this as we try to bring this to a close. This will not be the last discussion on GM food, the last presentation on GM foods. It must be an ongoing discussion. But we have, in terms of time, a limitation with regards to that. So, Prof, can you just quickly respond to what Prince Odo has said?
Yeah, thank you, Prince, for your lecture. Although I didn't quite get it all, but I think the first thing you said is, what is the model guiding the experiment? And I did say at the beginning that there is a biosafety protocol that guides everybody in the world. And those are the experiments you must perform and send their results with your dossier when you are applying for approval. It is a specific set of experiments so there is a model that guides the experiments. Then you asked a very good question about independent research to cross-check these foods. So the regulator assesses all the experiments and results submitted by the producer and satisfies itself that all is well. But it is a continuous thing. After a product has been released, scientists in the country, it is now our responsibility to continue to query, analyze, and interrogate the safety of this product. So some of us have done this. We are analyzing a BT cowpea to the extent that the funding of research in our country will allow. That is why I am telling our powers that be that it is embarrassing that there's so little that we can do, here. But we are making efforts with the little that we have. And you talked about protein evaluation, but I didn't quite get what you said about that. But the protein that is evaluated is the transgene, the protein encoded by the transgene. That is what you added. So that is the protein that is evaluated. So I don't know, the chairman can remind me if there's something.
Well, thank you, Prof. I think you have dealt with it ably. And clearly, in concluding, the operative word that you have used is continuous.
Yes.
The assessment, the evaluation, the research, public scrutiny, all that must continue.
Yes.
That we cannot run away from.
Yes.
Because absence of evidence is not evidence of absence.
Exactly.
In the field of food science and technology, when I was a graduate student, nobody would have talked about acrylamides in food. It's not that they were not there, but our detection methods were up to parts per million. Now we can go to parts per billion. Now we recognize that they are there. So, and that's the point being made. With the present level of knowledge, I think the point Professor Uzochukwu is making is that every evidence points to the fact that these foods are not generally unsafe, but the scrutiny must continue and the discussion must continue. And as she has said, for us in this part of the world, given the enormity of our problem, every technology must be on the table. We must be willing to evaluate, be prepared to use every technology. What we must not do is to come with a mindset that this technology is no good. There are evidences that we can point to with other technologies, whether they are talking about food irradiation or other technologies. So we must embrace every technology. We must take advantage of every technology. We must ourselves make sure that we are in the forefront of driving research in every technology. And that's where the point that Professor Uzochukwu again made becomes tragic for us as a nation, because we are not investing in what we should be investing in. So with that, for me, I should say that one of the concerns is, of course, the issue of crop biodiversity. For me, sustainable food security cannot just be based on a few crops, be it rice, wheat, maize, or soybeans, the big ones that we are promoting. It's a tragedy in this nation that our traditional crops that are indigenous to us, if we talk about cereal, sorghum, millet, acha, and the like, will always do better than maize in this country. But tragically, we have neglected those crops. And Prof will agree with me that from research, these crops are a lot more nutritious than maize, these 3 or 4 that I've mentioned, than maize, in terms of protein content, in terms of amino acid composition, they are superior. And we are finding out now that even in terms of health promoting food bioactives, they are rich in this. Whether are talking about biophenols, plant phenols of all sorts, polyphenols, flavonoids, alkaloids, name them. Our indigenous crops are rich in these health promoting components. Yet we have completely neglected them, and the whole world is promoting just 4 or 5 crops: rice, wheat, maize, soybeans. So really there are issues beyond the issue of safety of genetically modified food. So this discussion must continue, but we must remain open-minded. We must take advantage of whatever technology is available, and we must ensure that we are at the forefront of those technologies. So again, I want to thank Professor Uzochukwu very much. I want to thank all those who have asked questions. I want to thank Prince Oda. There must be such voices that we must listen to. So with that, I hand over to Professor Shweton to please bring this discussion to a close.
I apologize for coming in late. I had a network problem for almost 45 minutes, and so I joined in, I think, at half an hour. Sorry about that. Thank you very much, our distinguished Professor Ogugu Award, for your time for sharing this webinar. And we also like to thank, we have come to the end of this an enlightenment webinar, which is the webinar number 15 of the AfriScience Network, titled Assessing the Safety of Genetically Modified Food, by our esteemed Professor Sylvia Uzochukwu. She is the president of the Biotechnology Society of Nigeria, and also a professor of food science and biotechnology and biosafety expert at the Federal University, Oye Ekiti. On behalf of everyone present, I extend our heartfelt gratitude to Professor Uzochukwu for her invaluable insights and for sharing her expertise with us today. Her presentation has provided us with a deeper understanding of the safety assessment of GM foods, which is important for our ongoing and future research endeavors. We also wish to thank all participants for their active engagement and thoughtful questions, making this session truly interactive and productive. And as Professor Ogugu rightly said, the discussion and arguments on the GMO is unending, and I think we can continue these discussions on the platforms. Our platforms, we have 7 platforms from A to F, A to G, so they are always open for this kind of discussion. We also wish to thank all participants for their active engagement and thoughtful questions, and we look forward to more such informative sessions in the future. Thank you all for your time and passion, and have a great day ahead. Once again, my name is Aminu Shitu. I'm a staff member of the Faculty of Veterinary Medicine here at the Usman Danfodio University, Sokoto, Nigeria, and I'm one of the team members of the AfriScience Network. Thank you very much, sir.
Thank you, everybody. Thank you very much. I am much honored.